We can continue now with the presentation of the morning, and the first speaker of this afternoon is Professor Rahim Simsek. Just a few words before her lecture. Professor Rahim Simsek has got an MSc degree and PhD degree from ACTP University, Institute of Health Sciences, Pharmaceutical Chemistry Program. After she continued her postdoctoral studies at Virginia Commonwealth University School of Pharmacy, Department of Medicinal Chemistry in the USA. In 2004 and then in 2010, she was appointed as an associate professor and then full professor of ACTP University Faculty of Pharmacy. Professor Simsek was a visiting lecturer at the Turkish Republic of Northern Cyprus near Est University in 2018, and at the European University of Lefke in the Turkish Republic of Northern Cyprus in 2019. Her main research interests are design, synthesis, structural elucidation, and activity studies of new bioactive molecules, and Professor Simsek has worked as a researcher and director in many scientific research projects, and she has ongoing scientific projects. She published around 70 research articles in international refereed journals, and she was also awarded ACTP University 2008 and 2009 Academic Year Encouragement Award and 2008 Novartis Pharmaceutical and Medicinal Chemistry Drug Design and Development Research Award, 3rd place.
Thank you. Hello. Good afternoon to everyone, and thank you for a nice presentation. It is my pleasure to be here, and I have special thanks to Dr. Carradori and anti-cancer agents and letters in drug discovery and drug design and discoveries for kind invitation. Today, I'm going to talk about hexahydroquinone derivatives as potential inflammatory mediators, inhibitors. Actually, I am talking about our ongoing studies and the unpublished data. First, I'm going to talk about inflammation, and then one for dihydropyridine derivatives and hexahydroquinone derivatives, and then hexahydroquinolines and inflammation, and I'm going to talk about our future perspectives. As Dr. Gallorini said before, inflammation is the immune system's response to harmful stimuli such as pathogens, damaging cells, toxic compounds, and irradiation. In fact, inflammation is a defense mechanism. Some factors such as injury, obesity, smoking, stress, insulin resistance, and infection are the causes of inflammation. Inflammation can acute or chronic, and the acute inflammatory response eliminates possible injury and infection. Uncontrolled acute inflammation can become chronic and cause various diseases, chronic diseases such as cardiovascular diseases, neurological diseases, and so on. Triggering of the inflammatory response occurs through the pattern recognition receptors via pathogen-associated molecular pattern, PAMP, or damage-associated molecular patterns. PRRs include toll-like receptors, C-type lectin receptors, retinoic acid-inducible gene-like receptors, and NOD-like receptors. Here is the inflammatory response formation. Cell surface PRRs recognize damaging stimuli, and then inflammatory pathways are activated, inflammatory markers are released, and inflammatory cells accumulate at inflammation site, and the final effect will be inflammation. Primary inflammatory stimuli are interleukin-1 beta, interleukin-6, TNF-alpha, mediate inflammation by interacting with their respective receptors. Receptor activation triggers intracellular signaling pathways such as MAP kinase, NF-kappa B, and JAK-STAT. Pro- and anti-inflammatory cytokines are proteins that facilitate or inhibit inflammation. Inflammatory cytokines are interleukins, CSF, interferons, TNF-alpha, TGF-beta, chemokines, and so on. Although cytokines are molecules that govern the immune response, excessive inflammatory cytokines formation can cause some events from tissue damage to death. I think we don't have enough time, so I will not talk about too much inflammatory pathways and mediators. We are seeing some inflammatory pathways here, NF-kappa B, JAK-STAT, MET kinase, and we have some mediators here, TNF-alpha, interleukin-1 beta, and interleukin-6. TGF-beta is important. TGF-beta plays a role in maintaining tissue homeostasis. Adverse changes in the TGF-beta pathway are associated with many pathologies such as cancer, autoimmune diseases, fibrosis, cardiovascular diseases. TGF-beta is one of the most important factors, especially in cardiovascular diseases. Therefore, disturbances in TGF-beta signaling emerge as a new strategy for the treatment of cardiovascular diseases. Additionally, TGF-beta shows tumor suppressor effect in early stage of cancer, and in the late stage of cancer, TGF-beta contributes to malignant progression. So, pharmacological discoveries are invaluable for the treatment of TGF-dependent diseases. And another inflammatory pathway is oxidative stress pathway here. And then, okay. Here is the 1,4-dihydropyridine ring. This ring is actually a partially saturated form of pyridine ring, and this one is synthesized by Hansch in 1882 first. Hansch is German chemist. Hansch synthesized this compound by using alkyl acetoacetate, aldehyde, and ammonia. Today, nifedipine and many other drugs have 1,4-DHP structure. 1,4-DHP has some pharmacological activities, calcium channel modulator, potassium channel modulator, and so on. Yeah. Here is the hexahydroquinoline ring. Hexahydroquinoline is condensed analog of 1,4-DHP. Actually, condensation of 1,4-DHP with cyclohexane gives hexahydroquinoline rings. This one is partially saturated of quinoline ring. Hexahydroquinoline ring has some pharmacological activity. The most important one is calcium channel modulator activity. This ring system has also anti-cancer, antimicrobial, antioxidant, and anti-inflammatory activity. Okay. We have some calcium channel blockers, and the most important group is 1,4-DHPs. The first compound is nifedipine. Nifedipine was introduced to treatment in 1980s as an antihypertensive, and nifedipine and nicardipine is the first generation 1,4-DHPs. Then second generation comes. Third generation 1,4-DHPs have some superiority, and the last one is fourth generation. Cilnidipine, larcanidipine are the new drugs, and hexahydroquinolines are condensed analog of this ring system. One more time, and okay. We have many hexahydroquinoline compounds and investigated their calcium channel modulator activity. Our compound has small alkyl functions on second position of the ring, I mean methyl, ethyl, and third position has ester and amide structure. Aryl is attached to the fourth position. Aryls are substituted phenyl, naphthyl, indole, and so on. While our compound has 5-oxo structure, most of our compound have oxo structure on the fifth position of the ring. Also, we have synthesized sulfur dioxide compounds. Okay, sulfur dioxide compounds on this position. Some of our molecules has no substituent on 6th and 7th position, and the remaining molecules has dimethyl substituents on 6th or 7th position. Also, we got monomethyl or phenyl substituted hexahydroquinoline compounds on 7th position. We evaluated their calcium channel blocker activity. Then we published a paper according to our data, structure activity relationships for our compounds, and we have a review article about fused 1,4-DHPs, highly cited article, and also, we proved that our compounds has anti-nociceptive activity through calcium channels, and also we investigated their photochemical properties, and we have many publications on hexahydroquinones. Recently, some antihypertensive drugs having a 1,4-DHP ring were evaluated as an inhibitor for inflammation mediators. They have all common pharmacophores. Actually, the main ring has a 1,4-DHP ring, main structure. Small alkyl functions are attached to the 2nd and 6th position. 3rd and 5th position have ester function, and the disubstituted phenyl is attached to the 4th position of the ring. All of them are common pharmacophore for this drug, this anti-hypertensive drugs, and we are seeing common pharmacophore for drug. TGF-beta is important because it's involved in the mechanism of many diseases, especially cancers and cardiovascular diseases. So hexahydroquinolines can inhibit TGF-beta. Hexahydroquinone derivatives show a new mechanism of action for downregulation of TGF-beta receptor. And it is known that HHQs selectively inhibit TGF-beta receptor and inhibit intracellular signal mechanism. According to structure activity relationships on hexahydroquinolines as the TGF-beta inhibitors, 4th position phenyl ring provides selectivity, and the 3rd position ester function is important for activity. According to this information and our in silico studies, we have synthesized hexahydroquinoline compounds. In this study, we have 24 compounds having hexahydroquinoline structure as a main ring, and methyl is attached on to the 2nd position. 3rd position has various ester function, and the 4th position has phenyl ring, disubstituted phenyl. Substituents are phenyl and 3-fluoromethyl. Why fluorine as a substituent? Currently, 50% of the drugs in treatment has at least one fluorine atom. Fluorine makes the compounds more lipophilic and makes the compounds resistant to the biotransformation reaction. That's why we have selected fluorine substituents. We synthesized these compounds by using Hansch reaction of diketone compounds with appropriate alkyl acetoacetate and disubstituted benzaldehydes in the presence of ammonium acetate as a nitrogen source. This reaction gives our target compounds. We proved compounds structure by using spectral methods such as infrared spectroscopy, proton NMR, C13 NMR, HRMS, and X-ray analysis for some compounds. Okay. Within the biological activity studies, first we got cytotoxicity values for the compounds. C3C cell lines and HG2 cell lines were used with MTT assay. According to the MTT assay results, the less cytotoxic compounds were selected for further biological activity. The first experiment is ROS production in HG2 cell lines by using ELISA method. And 12 compounds were selected. According to our biological activity results, compound 1B reduced ROS formation and also interleukin-6 protein levels. Also, the same compound reduced interleukin-1B level, and compound 1B and 1C inhibit TNF-alpha production and also TGF-beta-1 production. Compound 1B was found to reduce protein levels in the inhibition test of NF-kappa B production. 5 compound decreased the 5 compound, including compound 1B again, decreased the TNF-alpha, TGF-beta-2 level, and according to our biological activity results, we have one lead compound, compound 1B. Here is the compound 1B structure. Compound has methyl on second position again, ethyl ester is here, and the aryl is attached to the fourth position. And the selected compounds show superiority to other ligands in terms of low binding energy and binding affinity in molecular docking studies. And this result is consistent with the result of in vitro experiments. In second project, again, we have synthesized hexahydroquinoline compounds, but this time, aryl has difluoromethoxy substance on para position. Some compounds has no substance on 6th and 7th position. Some of them have dimethyl substance. We obtained compounds by using Hansch method and proved their structure again by using spectral methods. And we have 15 compounds here. For biological activity studies, again, we have realized cytotoxicity experiments first time in C3T3 cell line by using MTT experiment, and less cytotoxic compounds were selected. We got 3 compounds. Our first experiment is ROS production, and according to ROS production experiments result, compound 2D has the lowest ROS production potential. And we have some results here. Compound 3E was observed to increase interleukin-1 alpha level. Compound 3B was found to reduce interleukin-10. And compound 2D and 3B decrease the TGF-beta levels, and all 3 compounds decrease the TNF-alpha levels. Okay. In order to determine the activity of compounds on the complement system, C3 and C9 inhibitor activities were determined in vitro. As you know, complement system is an effector mechanism of immunity in organism. According to the results, all 3 compounds decrease C3 protein levels. In case of C9 protein level, compound 3E provided a statistically significant decreasing, while the other 2 compounds provided a statistically significant insignificant decrease. And here is the molecular docking study for that compounds. Compound 3 is, active compound 3E has lower binding energy than other compounds, especially compound 2. This one is the less active compound, and the compound 3 has lower binding energy. It was observed that compound 3E had better affinity to the molecular target in the protein structure. So we talked about TGF-beta, and TGF-beta is important, especially in case of cancer and cardiovascular diseases. And we would like to see heterocyclic ring system effect on activity instead of small substance on phenyl ring. That's why we brought pyridine ring to the pyridine ring attached to the phenyl ring. We designed some compounds having this structure, hexahydroquinone structure again, and making in silico studies. At the end of our in silico studies, we saw that our compounds can be TGF-beta inhibitor. So we obtained our compounds by using Hansch method again, and evaluated their structure. Then we got biological activity studies. We used A549 and HepG2 cell lines. A549 human lung cancer, and HepG2 human liver cancer cell lines. They are known to have high TGF-beta receptor levels in cell culture studies. That's why we selected these two cell lines, and using flow cytometry analysis for cytotoxicity experiment, we got IC50 and IC80 values for the compounds. Compound 165, 170, I mean synthesized compounds didn't affect cell viability up to 10 micromolar concentration in both cell lines. When we look to IC50 values in A549 cell lines, compound 165 and 166 has lower IC50 values. I mean, these two compounds are more cytotoxic. In case of HepG2 cell lines, compound 169 has 601 nanomolar IC50 values. Then we got SMAD2 and pSMAD2 protein level value. SMAD2 is the protein, and it is the major receptor activated protein downstream of TGF-beta signaling. pSMAD2 is the phospho-SMAD2, phosphorylated form of the SMAD2. pSMAD2 is translocated into the nucleus to modulate the transcription of the target genes, and the pSMAD2 level indicates the activation of the TGF-beta pathway. We used reference beta-actin. Why? Actin is an important protein for cell motility, structure, and integrity. And we have also a commercial inhibitor as a positive control. In A549 cell lines, A549 cell lines, SMAD2 level remained the same. You are seeing here, and pSMAD2 protein was suppressed with commercial inhibitor, but synthesized compounds activate TGF-beta pathway. In case of HepG2 cell line, the commercial inhibitor didn't make any difference in the level of pSMAD2, and our compounds, synthesized compounds, activated the TGF-beta pathway by using pSMAD2. So we were expecting our compounds can be TGF-beta inhibitor, but we found that our compounds are TGF-beta activator. I mean, our compounds can bind to the TGF receptor, but they make activation on this receptor. The effect on pathway activation shows this substance can immunomodulate cancer cell. And okay. So we have synthesized 5-oxohexahydroquinoline compounds as a potential inhibitor on inflammation pathways and mediators. According to our results, biological activity experiments results, if the compound has small substance on phenyl ring, this type of compounds have the ability to inhibit inflammation on mediators to some extent. If the compound has a heterocyclic ring instead of small substance on phenyl ring, this type of compounds show TGF-beta activation. We cannot say something about ester function as a structure-activity relationship. And what we are gonna do in future, we would like to get more selective and more effective compounds and inhibit inflammatory pathways and mediators, especially TGF-beta. And we would like to determine our compounds experimental physicochemical and pharmacokinetic parameters. And we would like to make in vivo studies on diseases models. And if we have obtained a promising 5-oxohexahydroquinoline derivative, of course, we will be happy. And Gökhan was my PhD student. He was graduated in 2022. He synthesized first and third project compounds. Ezgi is my PhD student, and she synthesized second project compounds. Biological activity studies were realized in Esendal Laboratory and Erkekoğlu Research Group in Hacettepe University. And thank you for your attention. I hope I completed just in time. And greetings from Ankara.
Thank you so much for this amazing journey in a different aspect of chemistry, and especially for the biological results, which are very, very interesting and promising. I think that this field of research is very important, and you must continue on this pathway, because it is really, really interesting also for us. Thank you so much. You stayed within your prefixed time, so perfect. And sorry again to be postponed after the break, the lunch break. Okay.
No problem. Online world, you know.
Thank you so much for your contribution. Thank you.
Thank you.
Now it's time of another speaker, also a friend of mine who worked with me at the safety department when I was born. It's the turn of Professor Giovanna Poce. Giovanna Poce obtained her PhD in pharmaceutical sciences in 2008 from Sapienza University of Rome. Then she did a postdoc at the Department of Chemistry of the Oxford University in 2008, and then the Harvard School of Public Health in 2010. In 2013, she came back to the Sapienza University as an assistant professor at the Department of Chemistry and Technology of Drug, where now she's working as an associate professor in medicinal chemistry. She is an experienced medicinal chemist, and her research focuses on the design and development of small molecules active against Mycobacterium tuberculosis, hitting novel targets such as MMPL3, iron chelation, tryptophan biosynthetic pathway, but also malaria transmission blocking and antiviral compounds. The title of her lecture is Simple Chemistries as Enabling Tool for Advancing Antimicrobial Research.
Thank you, Simone, for your kind introduction and for inviting me to this interesting webinar. Today's talk is going to be about our approach on developing new possible drugs working against microbes. And this is because there is an urgent need in developing new anti-infectives against antimicrobial resistance, especially because the pipeline of drugs under development is very weak. Indeed, the WHO identified 32 antibiotics in 2019 in clinical development that address a list of priority pathogens, but only 6 were classified as innovative. My research group at Sapienza is involved in different medchem projects, and several of them are about developing anti-infective compounds. So for today, I want to focus my talk about the development of anti-TB drugs, and especially 2 classes of active compounds, one as MMPL3 inhibitors and the other one working on the alteration of tryptophan biosynthesis in Mycobacterium tuberculosis. And the other class of compounds are active against enteroviruses. So basically, following the unmet need of developing new drugs, a new anti-TB drug working with a different mode of action for avoiding cross-resistance with the current therapy, we identified a new class of antimicrobacterial agents acting against MMPL3. MMPL3 is a mycobacterium membrane protein large, and is involved in the construction of the barrier in mycobacteria. So we have identified a heat compound starting from a phenotypic screening. From the identification of the heat, of course, we moved ahead with a heat to lead campaign, and this campaign brought by an improved heat called BM635. BM635 proved to be very active both in vitro and in vivo, even though it showed some liabilities in the DMPK profile. So in the fight against poor water solubility, poor metabolic stability, and hERG activity, we decided to replace the central pyrrole core with a pyrazole 1, and we obtained a new series of active pyrazole compounds, and some of them proved to be as active as the heat compound, but with improved DMPK profile. Okay, so with this in our hands, we joined the Tuberculosis Drug Accelerator Program, which is a consortium of both industrial companies and academic institutions. And these people in this consortium share data and compounds with the aim of identifying new anti-TB drug. And the consortium believes that MMPL3 inhibitors can synergize with the current therapy in reducing the time of current treatment. And this because SQ109, one potent MMPL3 inhibitor that passed phase 2 clinical trials, proved to be active in reducing the time of the current treatment. So we are now focusing our attention on improving the most active compound, the most active pyrazole derivative, BM859, because it proved good activity in vitro, good solubility, and it also proved to be active in vivo on an acute model of a TB infection, but at 200 milligrams per kilo. So our aim is now to reduce the in vivo effective dose by improving the PK profile. So then we started with analyzing the possible metabolism of fate of BM859, and we found two main metabolites. One is given by the demethylation of this methoxy group on the piperidine, and the other one is given by the hydroxylation of one of the aromatic rings. Therefore, we decided to remove these soft spots, metabolic soft spots in BM859, and we are now synthesizing and improving the series, introducing different substituents of the soft spots, different substituents that are known to be more metabolically stable in vivo. So the other class of anti-TB compounds we are working on are working through the alteration of tryptophan biosynthesis. Indeed, several studies demonstrated the essentiality of many of the enzymes involved in biosynthetic pathways in pathogens that live in intracellular environment like Mycobacterium tuberculosis. So basically, by starving so basically the immune system tries to counteract the infection by starving the bug of nutrients of one of these amino acids. And therefore, the bug counteracts the immune system by making amino acids itself. So basically, by targeting one of these biosynthetic pathway could synergize the immune system to, to block infection. Moreover, these amino acids are essentials. Therefore, enzymes lack orthologs in humans, and therefore inhibitors of these pathways could be selective inhibitors. Nowadays, there is a growing interest in studying mycobacterial tryptophan biosynthesis and its critical role in counteracting the immune response upon infection, upon MTB infection. Indeed, a fluoroantranilate compound called 6-FABA proved to help the immune system against TB infection, and this 6-FABA and its ethyl ester proved to be active in vitro and proved also to act through the biosynthetic tryptophan biosynthesis, because the BCG growth was rescued from 6-FABA activity by adding tryptophan to the culture. So 6-FABA and its ethyl ester proved also to be active in vivo by reducing bacterial load on in infected mouse spleens. So with this intriguing data, we decided to make a series of these FABA derivatives. And we focused our attention on modifying the carboxylic function. And this is because 6-FABA and the ethyl ester showed some toxicity in vivo. And we know that mammalian human toxicity is related, the carboxylic function is toxic in human and mammals because of the rearrangement of some acyl glucuronides. So we decided to focus on removing this carboxylic function, and we replaced it with hydrosamic acid or ester, or with some bioisosters like oxadiazole or tetrazoole. Or we decided to replace the ester function of the ethyl ester derivative with the more metabolically stable amide function or hydrazides. Finally, we also made a small subset of compound of trifluoromethylamines, and this because this substituent grants the same behavior as amide bonding, is more water soluble, and is more stable, metabolically stable. So within all the synthesized compounds, only the hydrazides proved to be active, and especially these three ones. They showed good activity when tested in different media. So our concern was about the hydrazide, the possible toxicity of hydrazides. Indeed, as it's well documented for isoniazid, hydrazides can lead to active radical species in vivo. But to our satisfaction, the active hydrazides didn't show cytotoxicity when tested on viral cells. So to prove that this Faba compound could act by inhibiting tryptophan biosynthesis, we started some biological experiments. And we started by testing the activity of the Faba derivatives by adding in the media either tryptophan or some of the most important precursors like indole, antranilate, and chorismate. And we noticed that by adding tryptophan, indole, and antranilate, BCG growth was rescued from Faba inhibition. This told us that they might act by alteration of this biosynthetic pathway. We went ahead and generated resistant mutants to one of the Faba compound, and we sequenced the genome and found point mutations in 4 of the enzymes involved in tryptophan biosynthesis. So the fact that we are lacking one mutational hotspot, and we get mutation in different enzymes involved in the biosynthetic pathways, it tells us that this mutation might block the flux of tryptophan precursor through the pathway, instead of blocking the direct interaction of the compound to its possible target. And this is seen very often in antimetabolites that basically poison a pathway downstream and behave like altered substrate in the pathway. So finally, to determine the biochemical interaction between the Faba compound and these enzymes, we used CRISP interference to generate inducible repression of these 4 genes, TRPB, TRPD, TRPC, TRPC, and TRPE. And when comparing with the CRISP empty vector control, we could see that activity of the repression of TRPB and TRPD desynthesized Mycobacterium tuberculosis BCG to Faba activity, and this supporting the idea that these compounds act as alterators of the tryptophan biosynthesis. Of course, the exact mode of action needs to be further elucidated, and we are still working on that through isotopic tracing of the active hydrazides. We are also making more analogs especially removing this NH2 here, this substituent, because we still didn't investigate the importance of this substituent on the activity of this class of compounds. Finally, the other project I want to talk about is on the development of compounds that act as a capsid, binders, as antiviral against enteroviruses. So enteroviruses are small non-enveloped viruses, and they affect millions of humans yearly, and usually they cause mild infections, but are also associated with serious illness like diabetes, heart attack, acute stroke, and hand and foot and mouth disease. We decided to focus on two serotypes, Coxsackie virus A9 and B3. So Coxsackie virus A9 has been associated with outbreaks of meningitis and was found to cause hepatitis, while Coxsackie virus B3 was associated with myocarditis, pancreatitis, as well as meningitis. So we started our project by cell-based screening against CVA9 and CVB3 of an in-house chemical library of 200 compounds of different chemical scaffolds. So we identified molecules that were active at 10 micromolar, and then we went lower in the concentration, and we identified two hit compounds, Cl212 and Cl213. They both proved to be their antiviral activity getting very high cell viability, but they proved to be selective against CVA9 and inactive against CVB3. They showed a very good EC50 and also good cytotoxicity. So to get an idea on the mechanism of action of this compound class, we did a time of addition assay. So basically it works by studying the cell viability at different concentrations and at different time points, and we noticed that they were both able to reduce viral replication, either added before the inoculum or 1 hour after the inoculum. So telling us that they were working at the very beginning of the infection. We went ahead and did other testing, and we measured, we did this test that is called encoding assay, and we noticed that by incubating cells with Cl212 and Cl213, we noticed that they stabilized and prevented the encoding of the virus. And this was proved also by images got by transmission electron microscopy. So they behave like stabilizing the particles, and so therefore viruses cannot release their genome in the host. Normally this is a behavior, this class of compounds is normally called capsid binders. So we are now following two parallel programs. One is on continuing studying the mechanism of action, and the other one is on improving the compound series. So for making more analogs, the first step was to build up SAR, and we built up initial SAR by analyzing the initial chemical library. That was the initial chemical library for searching for analogs that could share some similarities with the two hit compounds. And we were able to build up some preliminary SARs, and we found that the phenyl benzamide core is essential for the activity, as well as the N-methylpiperazine here, especially at the para position, and especially because when moved to other position, it didn't work at all, and when was replaced with morpholin or thiomorpholin or pyrazose analogs, didn't work at all either. So we are now going ahead and made more derivatives, and the first step the first step of the project consisted in replacing the amide bond with an amine one in the two with maintaining the other substituents, then changing the substituents on this phenyl ring, or changing the substitution on the piperazine. Surprisingly, only two compounds, the amine analogs of the hits, proved to be active, and they also proved to be more active than the hit compound, telling us that a more basic characteristic enhance the antimicrobacterial activity. So we, as I said, starting from this data, we are making more amine analogs, but I do not have data at the moment. And we are also using cryo-electron microscopy to see how these compounds fit into the VP1 pocket of the CVA9. And from the picture, we could see that the molecule fits well into the density, and it's different from the lipid factor, which normally occupies this pocket. So based on this structure we got, we are also applying docking analysis to get more information, and so combining all this data, we are designing and preparing new derivatives. With this, I would like to conclude, and before that, I would like to thank all the groups that are involved in these projects. So the group at Sapienza, and then the group of Professor Cibale at the University of Cape Town, Professor of Newstead at the Department of Biochemistry in Oxford, Professor Tanya Parish from the School of Medicine, and Dr. Elena Boshoff from the NIH for the MMPL3 inhibitors project. Then the group of Professor Eric Rubin at the Harvard School of Public Health, the group of Thomas Jorger at the Texas University, and the group of Professor DeLogo at the University of Cagliari for the project of the tryptophan biosynthesis inhibitors. And finally, the group of Professor Barpo Marjomaki and Sarah Butcher from the University of Jyväskylä and Helsinki for the antiviral compound project. And all of you, of course, for your attention.
Thank you very much for your presentation. I'm sorry that we have no time for questions, because we are trying to save time in order not to finish overnight. But thank you so much for this specific overview of targets and exploitable targets that we didn't deal with this morning. So thank you so much. Now it's the time of the next speaker. He's a rising star of medicinal chemistry in Italy, Dr. Andrea Angeli, graduated in chemistry and pharmaceutical technology in 2014 at the University of Florence, where in 2019 he obtained his PhD in medicinal chemistry. Currently, he is a postdoctoral researcher in the research group of Professor Claudius Supuran, and his main research activities are related to the synthesis and biological evaluation of small molecules as carbonic anhydrase inhibitors for the treatment and diagnosis of relevant human pathologies. He participated in several national projects, and despite his young age, he's co-author of more than 250 papers in peer-reviewed journals and 2 patents. So I'm very glad to present the next speaker, who is Andrea Angeli.
So thank you, Professor Carradori, for your kind presentation. Also, thank you to Professor Cacciatore. Today, talk about carbonic anhydrase inhibition as promising neuropathic pain relief agent. So pain is defined as unpleasant sensory and emotional experience associated with actual or potential tissue damage. There is several kinds of pain and is divided by different factors. One of them we can classify based on its etiology. From one side, we found nociceptive pain caused by tissue damage and well treated by non-steroidal anti-inflammatory drug or opioid drug for more severe form. On the other side, we can find neuropathic pain is caused by nerve damage, and today there are no valid pharmacological treatment. This kind of pain is associated mainly as one side effect of oncological chemotherapy, in particular for platinum-based drug, such as cisplatin and oxaliplatin. And recently was found their neurotoxicity is mainly affect the dorsal root ganglion neurons. As I mentioned before, there are no effective analgesic treatment for this kind of pain, so it's very important to understand the mechanism behind the pathogenesis of this pain in order to find novel biological target to treat the neuropathic pain. In the last decade, several research groups found one promising target, carbonic anhydrase, is found involved with different mechanisms in the pathogenesis of neuropathic pain. What is it? Carbonic anhydrase are ubiquitous metal enzyme, are zinc enzyme mainly, and present in all kingdom life. All the family catalyze the interconversion of carbon dioxide and water in carbonic acid, but this simple reaction is at the basis of many physio and pathological events. In the human, we can find 15 alpha isoforms that are different tissue and cellular distribution. To date, carbonic anhydrase inhibitors are well-known chemical agent for the treatment of several diseases, from glaucoma, epilepsy, more recently as anti-tumor agent. But how carbonic anhydrase is involved in neuropathic pain? We can find 2 different mechanisms. One central is found involved in a GABAergic pathway, where in a chronic pain condition, we found the co-transporter CC2 decrease its activity, leading an imbalance between bicarbonate and chlorine anion. This leads an extra depolarization of the membrane, and one possible mechanism to treat this, to resolve this imbalance, we can use carbonic anhydrase inhibitor to reduce the bicarbonate anion production to restore this equilibrium. On peripheral mechanism, more recently was found platinum drug make several complexes with hemoglobin, a protein that acts as a proton buffer in intracellular dorsal root ganglion neurons, and this complex leading an intracellular acidification. This acidification has a consequence many protein dysfunction, among them the family of TRP protein. So also in this case, the employment of carbonic anhydrase inhibitors can reduce the proton beam production and restore the optimum pH. Keep in mind this consideration. My research group started in 2015 to study novel carbonic anhydrase inhibitors has a potential neuropathic pain agent. In particular, start to synthesize ether compound with benzene sulfonamide moiety. This moiety is essential for the carbonic anhydrase inhibition. Among this series was found one compound here reported with a very good inhibition potency against several isoforms, in particular for the ubiquitous isoform CA2 and the brain-associated CA7, they are the most involved in neuropathic pain. This compound is tested in vivo in axial implanted neuropathic pain model and compared with the reference drug acetazolamide, a well-known carbonic anhydrase inhibitor. So both compounds showed a rapid onset of pain relief effect with a maximum peak around 50 minutes, but acetazolamide showed also a rapid offset. On the other hand, the novel compound lasting the effect for 1 hour. So the novel compound are a very good pain modulator agent. And here start my research. So how is it possible to improve this effect? We moved on the synthesis and the evaluation of carbonic anhydrase inhibitors with antioxidant property. To obtain this compound, we made a very simple isosteric replacement of oxygen atom with selenium. Selenium is a well-known element with antioxidant property to counteract the oxidative stress related to mitochondrial damage caused by platinum drug. We have a large series of selenide bearing benzene sulfonamide, and among them we selected 3 compounds, the best 3 compounds with high potency against the 2 isoforms involved in neuropathic pain, CA2 and CA7, with nanomolar and subnanomolar KEI inhibition. Among these 3 compounds, we can find the last one, 7A, that showed a very strong pain relief effect with a rapid onset, but this time all compounds showed a quite different pain relief effect with a maximum around 30 minutes and rapidly decrease until 1 hour. The last compound but showed a very strong pain relief effect because the dose used is 3 times less than the other and the reference acetazolamide. So also selenium can be used as a pain relief agent, but this enhancement of effect is caused by antioxidant property, or this antioxidant property can affect anti-tumor effect of platinum drug. To answer this question, we try to synthesize a polychalcogenide compound with sulfur, selenium, and tellurium moiety. Also, tellurium is a well-known element with high redox property. And we try to investigate if co-administration of our antioxidant property with platinum drug can affect their anti-tumor property. we selected 3 compounds with good inhibition potency against several CA isoform and are tested in vitro against one tumor cell line, one breast adenocarcinoma line, and before we tested alone, and the first 2 compounds with selenium and sulfur moiety don't show any cytotoxic effect. On the other hand, the tellurium moiety we detected EC50 of around 125 micromolar. At this point, we need to find a dose that are not toxic for our compound, but also for cisplatinum drug, and we found 100 micromolar concentration is a subcytotoxic dose for our experiment. And when treated platinum drug with our compound, we see an enhancement of anti-tumor effect for both 3 compounds. So our compound not affected the anti-tumor property of cisplatin, but increase, we have a boosting of this effect, and this is a very important feature because is a possible novel strategy to use carbonic anhydrase inhibitors to enhance the anti-tumor effect, and so we can reduce the anti-tumor dose of platinum drug and as a consequence reduce the side effect. Moving on the same in vivo model, we replace the tellurium compound with a non-cytotoxic compound with 2 selenium atoms, and this compound showed the best neuropathic pain relief effect. All 3 compounds have a rapid onset of pain relief, as previously seen for the other selenide compound, but this time compound 5E showed at 30 minutes the maximum peak and revert completely the neuropathic pain effect. So it's a very potent agent against this pathology. In addition, we also increase the time over 1 hour for the pain relief. At this point, finally, we moved on specific polypharmacological compound. We start to target carbonic anhydrase enzyme and another well-known target for pain management, the TRPV1 receptor. And we try to obtain compound able to target contemporarily these 2 proteins. To obtain this compound, we start from a lead structure of TRPV1 antagonist and installing the sulfonamide moiety on 2 positions, one on phenyl ring and another on pyridine ring, in order to evaluate possible different interaction with both targets. Here reported the synthesis of this large series for both enantiomers, but the reaction is very simple and I will skip for time reasons. So we have in hand a large library of compound, and all these series are tested against several CA isoforms, including CA2 and CA7, they mainly involved in neuropathic pain. And we can see very interesting feature for all the series. We don't see any difference between the enantiomers against CA2. So in vitro, don't see the stereocenter play a pivotal role in the potency. But the installation of sulfonamide in both positions switch the effect on TRPV1 receptor from antagonism to agonism. So we have in hand agonism compound with carbonic anhydrase inhibition. Also for the TRPV1 receptor, we don't see any difference between the enantiomers. So for these reasons, we move on the study why the stereocenter now play a pivotal role in carbonic anhydrase, in particular for isoform 2, and we study by protein X-ray crystallography two compounds, a couple of enantiomers with similar carbonic anhydrase inhibition for CA2, and obtaining the complex for both compound. And how you can see, both compounds show the same binding mode, interacting with the zinc ion. These compounds are located in the same hydrophobic task depicted in red, but the very interesting point is the stereocenter play a pivotal role in the interaction with the protein. Indeed, the tail of the two compounds showed an opposite orientation, so the stereocenter is important for the interaction with the protein, even if the effect on the potency is the same. For this reason, when we moved in vivo neuropathic pain study, we selected different compound with different feature. The first two showed high potency against CA2 and CA7, but not show any agonism effect on TRPV1, and this reflect in a in vivo model with a late pain relief effect with a maximum of 45 minutes and decrease around 75 minutes. After, we selected two compounds with both activity, so carbonic anhydrase inhibition and TRPV1 agonism, but a different location of sulfonamide moiety, one on pyridine ring and another on phenyl ring, and benzene sulfonamide showed the best effect. So the installation of sulfonamide on pyridine ring decreased notably the neuropathic pain effect. On the other hand, phenyl ring showed a rapid onset and lasting this effect for over 1 hour. Finally, we selected a couple of enantiomers with similar carbonic anhydrase inhibition and TRPV1 agonism effect, and we evaluate if the same in vitro potency reflect also in vivo experiment, but not showed the same effect in vivo. So the enantiomer are the best with a rapid onset, lasting and reverting for 15 minutes from 30 minutes to 45 minutes, the effect, the pain relief effect, and lasting for over 1 hour. So, in conclusion, we can see as carbonic anhydrase inhibitors is a promising biological target to treat this kind of pain that are at the moment lack of effective pharmacological agent. We can employ organochalcogenide carbonic anhydrase, so with antioxidant property to increase this effect, and but also can increase the effect, the anti-tumor of platinum drug, and this has a consequence, the possibility to reduce the dose of anti-tumor agent, so the side effect. And finally, one promising a way to find potent and with a very lasting effect of pain relief effect, targeting carbonic anhydrase enzyme and TRPV1 receptor. Thank you for kind attention.
Here I am. Thank you so much, Andrea, for your nice presentation. Another overview of the world of carbonic anhydrases. And thank you so much. I see you in Naples for the next conference of carbonic anhydrases, where we are also involved together. Thank you so much. Now, the last speaker of this session and also of today. I would like to introduce you Professor Alexander Timoshenko. Professor Alexander Timoshenko is an assistant professor at the University of Western Ontario, Department of Biology in Canada. He earned all his degrees at the Belarusian State University and completed his postdoctoral and visiting scholar research in Germany and in Canada. The main areas of his research expertise include biological activity of plant and animal lectins, cell biology of galactins and cell aggregation, molecular mechanisms of cellular stress responses, reactive oxygen species and redox regulation, cancer biology, lymphadenogenic factors, innate immunity and neutrophils. His current research explores the role of galactins in regulating cell stemness and differentiation. Professor Timoshenko, now we will provide you the ability to share your screen. And the title of his presentation is The Role of Tissue-Specific Galactins as Tumor Suppressors.
So basically, I'm far away from Italy right now, and I'm talking from Canada. And I would like to thank Dr. Carradori and the organizing committee for inviting me to talk about galactins and the recent research with tissue-specific galactins in my lab. I don't see the rest of my figures on this slide, but first things what I would like to mention to introduce the galactins, and I mentioned that these are specific type of glycan-binding proteins which are encoded by 16 different type of genes, and they are characterized by the fact what I don't see on my screen, and I'm not sure that you see it. They have a specific carbohydrate. They share a specific carbohydrate binding domain, which allows them to bind beta-galactoside containing glycans. They share significant similarities in their carbohydrate recognition domains, which exist as a beta sandwich of 5 and 6 beta strands are responsible for binding the glycans. What is important about these proteins that they actually multifunctional proteins, and I hope next slide show us what's happening with them. They were found out to participate in the many different type of cellular processes, and actually, as you can see here, any type of these fundamental cell processes, what we know, there is information in the literature that they different type of galactins participate in their regulation. For example, cell adhesion, apoptosis, autophagy, differentiation, migration, cell growth. There is also a lot of information about the galactins and their association with different type of diseases, including this different type of blood diseases, cardiovascular disease, for example, cancer, and brain. So if we look at the expression, if you look at the expression of galactins in the human tissues, and human cells and tissue express 12 different galactins out of 16. You can notice here that there is a huge variations in the tissue-specific expression galactins. Some galactins, like for example GAL1, 3, 8, 9, they have a low tissue specificity, while galactins like 4, 7, and 10, they are tissue-specific. Even more, if I can say, like tissue specificity associated with galactins with the high numbers, which include like GAL12, which is specific for adipocytes tissues, and galactin 13 to 16, which is a specific for placenta. So the expression of the galactins, of course, obviously is regulated by different type of transcription factors, and based on the bioinformatic analysis using the PROMA software, it's obvious that also many transcription factors are predicted to regulate the galactins, but at the same time, there are a lot of transcription factors which don't overlap in terms of regulation of different galactins, which may explain the different type of tissue-specific patterns excretion of galactins in cell and tissues. So, and now to be closer to the topic of galactins in cancer, I would like to mention that there is like a huge amount of publications available of PubMed, and out of this more than 10,000 publications, 1/3 is related to the cancer and cancer problem, related to the cancers in different aspects of the cancer biology. If a most common view about the galactins that the expression is upregulated in cancer tissues, and really the use of like inhibitors of galectin-1 and GAL3 actually has a positive application in vitro in terms of like to inhibit the tumor growth and tumor progression, tumor cells. However, if we look at the hallmarks of the cancer, which are defined, as we know, in the past by Hannan and Weinberg, you can see here that different galactins can participate, can have a different relation to this hallmark of the cancer, which include like a promotion of cancer-related markers, but in the inhibition. And indeed, there are some galactins, in particular 478, which are claimed as tumor suppressors. So, considering all this type of relation of galactins to the cancer biology, of course, many different type of inhibitors were designed and available. And they include like drugs which represent carbohydrate inhibitors here, and also allosteric inhibitors. All these inhibitors, what they do, they just designed to inhibit the carbohydrate binding activity of galactins. However, the application was not always lead to the expected outcome in terms of like treatments of cancer. And here, by looking at this slide, what I would like you to pay attention that even there are so many type of inhibitors, many of them, they are not exactly specific to specific type of galactins. And there is overlaps in the binding of these inhibitors with different type of galactins, or if we are talking about them, let's say allosteric inhibitors, the galactins can serve not only as a glycan binding proteins, but all inside of the cells, they can work in the glycan independent manner. But these inhibitors, they very often they don't address this ability of to affect the glycan independent functions of galactins. So with these messages, like if still the galactin inhibitors were used for many clinical trials in combination with other antioxidant drugs or its own. But so far, the results of these clinical trials is pretty much didn't go beyond the initial phases of the trials, and none of the galactin inhibitors were achieved the point when they can be used to treat cancer. And especially this message, what I would like to show here, this message also relates to, if you look at the cost of the stocks of companies which produce these inhibitors, like for example, Galectin Therapeutics and Galecto, you know, the stock prices dropped down significantly over the last years, even what indicates that the success of these inhibitors is very challenging. I'm not criticizing these companies. These are absolutely great companies, and they do a great job to design these inhibitors. But this is not this really challenging exercise due to, again, those few things what I already mentioned. The first of all, the ability of galactans work in the glycan-dependent, glycan-independent manner, independent manners, but also due to the fact that galactans are soluble proteins and they can work inside and outside of the cells. And their functions outside inside cells are very different. And because of that, there is a need to consider more carefully and develop different paradigms how to understand better how to use galactans in terms of like for cancer therapy as anti-cancer drugs or for anti-cancer research, considering additional approaches and considering the cell biology of galactans. And what we are doing now in my lab, and what I would like to pay attention in this context, the ability of galactans to induce and be associated to regulate the cellular differentiation. So on this slide, what we see here, I put here a different type of cells which differentiation of which is associated with the changes in the expression of galactans. And as you can see here, many differentiation of many lineages associated with the upregulation of galactans. So, and if we have a case of upregulation and we have differentiated cells, so we can kind of guess that these type of molecules maybe not we don't need to inhibit them because considering the concept of differential therapy of cancer. However, of course, there are some cases where we see a downregulation of galactans. So altogether, what I want to show here that tissue-specific signature of galactan expression suggests that galactan network may act as a through a mechanism which controlling cellular differentiation, and it should be taken into account when we are trying to change the activity of the level of galactans in cells and doing any type of therapy. Moreover, I would like to mention that there is a lot of information in the literature with recombinant galactans, and many of them were found to induce differentiation of different types of the cells. Okay. However, again, there are cases of inhibition. That's why there is still unclear balance between the way how galactans can stimulate or inhibit cellular differentiation. This what we are trying to do in my lab, we would like to consider the different contribution of galactans to the regulation of process of cellular differentiation, considering tissue-specific galactans, because the general message is that those galactans which are has a low tissue-specific expression, they usually are regulated under the in transformed cells. But this is opposite often to the galactans with to the tissue-specific galactans. So what we are doing in my lab, we are focusing on two very highly specific tissue-specific galactans, GAL12 and GAL16, which using several cell culture models using the cancer cells which represent acute myeloid leukemia, breast cancer, choriocarcinoma, and glioblastoma, and which can be induced by these drugs like retinoic acid, all-trust retinoic acid on DMSO, or CMP analogs to change the galactan expression profiles in these cells. So, first, I would like to start with GAL16. GAL16 it has the highest number actually, and this is the only galactan which is actually less studied among other galactans. It's a gene encoding these galactans is localized on the chromosome 19, and very little information available on function of this galactan in cell and tissues, and as such, this galectin was described. Also, this galectin was described about like more than about 15 years ago in 2009 by the group of Tan. But since then, there is a very limited information about this galectin. There is no information even about when will you read the review on galectins. Many current reviews, they don't specify this galectin as a part of the galectins group of proteins. And one of the reasons for that is that this galectin is not detected yet, and there is no articles or studies which would detect this galectin as a protein level in cancer cells, in tissues or cells. The only this galectin, the recombinant galectins was produced, and you see the structure of the galectin here. And this is only what is known about the this galectin, the protein level. No, at least it shows us that is a galectins with has a prototype galectins here. What we did, we used different type of bioinformatic analysis, in particular the data sets available at the Human Protein Atlas, and we find out that this galectin is really highly tissue specific and expresses a high level in a very limited number of tissues, which include placenta, retina, and brain tissues. So we, with this type of information, we decided to look at the expression of this galectin first in the placental tissue, in the cells which cancer cells which can be differentiated to the trophoblast-like cells represented in placental tissues. What you see here on this slide, we designed, of course, this specific primers which allow us to test the expression of this galectin in two cell lines, BO and JAK3 cells, which are choriocarcinoma cell lines. And our results were very interesting because we find out that also the basal level of the expression of this galectin in both cell lines is very low, but induction of differentiation of these cells to the trophoblastic cells leads to the significant upregulation and easy detection of this Gal-16 in differentiated cells. These cells are differentiated, trophoblastic differentiation of these cells is confirmed by the parallel upregulation, expression of chorionic gonadotropin, a biomarker for trophoblastic cells, what you see here. So to look at the potential mechanism which can regulate the expression of Gal-16 in these cells, we used a set of different inhibitors which are related to the transcription factors, which potentially can involve in the regulation of these galectins based on our bioinformatic analysis, as well as inhibitors of CMP-dependent pathways, because CMP is a like common inducer of this type of differentiation. We use a panel of 7 inhibitors, and out of this panel, we find out that only 2 inhibitors or 2 drugs affected the expression of galectins in the differentiated galectin 16 in differentiated cells. This is inhibitors of p38 MAP kinase pathway, and this EPAC inhibitors or stimulator. Actually, it induces the upregulation. No, basically, the conclusion what we may conclude at this point that the signaling pathway which is mediate the upregulation of Gal-16 there are 2 branches here which are functional. PK dependent branch, but it's kind of not directly, it's through p38 MAP kinase, and also PK independent pathway. So and more things what we did to understand the better the functional role of this galectin and show its relation to the differentiation of these cells. We generated the CRISPR-Cas9 Gal-16 knockout JAK3 cells and look at their differentiation. And the findings very, very interesting and impressive for us because once we knock out Gal-16, these cells lost the ability to be differentiated based on the expression of CGB, chorionic gonadotropin in these cells, and based on the expression actually chorionic gonadotropin in the both protein and gene level, and based on the inhibition of based on the absence of inhibition of growth of the cells in the place of the CMP, because once the CMP cells differentiated, normally their growth rate is inhibited, but knockout cells, they show no differences in the effect of their growth rate in response to CMP. So this data allow us to make very important conclusions that Gal-16 actually is required for trophoblastic differentiation of JAK3 cells. So next, as remember, I said that this galectin also based on the bioinformatic analysis is expressed in the brain tissues, and we used U87 glioblastoma cell lines to look at the expression of this galectin in these cells. So these cells actually can be differentiated into different type of brain-like cells, astrocyte-like cells using CMP analogs or sodium butyrate induced to the neural cells. And what we find out that once we induce this type of differentiation, we notice, yes, these cells were differentiated and they show all these markers of differentiation. Doesn't matter what type of inducers we use here. We see increase in the number of the cells with processes. We see a drop in ogluconylation or number of oglucosylated proteins in cells is a sign of the differentiation. And at the same time, here we notice the significant upregulation of Gal-16 in response to the CMP analogs, but not in response to the sodium butyrate. Basically, what does it mean? That the Gal-16 is part of the functional activity of astrocytes and but not neural cells, and it also indicates that most likely Gal-16, this data, what available on bioinformatic analysis, they represent the level of Gal-16 in normal brain tissues, which are associated with astrocytic cells, astrocytes. So, and now I want to move to the Gal-12, which is other various tissue-specific galectin, which is expressed mostly in adipocyte tissues and breast tissues and bone marrow cells originated from bone marrow, for example, neutrophils. This is, you see, this again, the data extracted from the Human Protein Atlas. They show kind of this RNA expression in very limited number of the tissues. Gal-16 is a little bit complex gene because there are several splicing variants of this protein with different biological functions. And another little bit question about this galectin, that crystal structure is not resolved yet, but predicting structure showed us that this galectin is a part of the tandem repeat galectins, which has a 2 carbohydrate binding domain you see here. So the very comprehensive work from the Futong Liu lab showed that this galectin is an important regulator of differentiation of adipocyte and sebacytes. Okay, it's also involved in the regulation and modulates this cell plasticity of macrophages, the conversion from M1 to M2 phenotypes, and also involved in the regulation of differentiation of granulocytes. Very important feature of this galectin in comparison with all other galectins, as far as we know at this time, that by its association or association with the lipid droplets. This galectin actually positively associated with lipogenesis inside of the cells. It's localized on the surface of the lipid droplets. So to study this galectin, we use 2 different cell models, but first we look at the changes in the expression of this Gal-12 in the breast cancer. In breast cancer using the TCGA, the Cancer Genome Atlas database. The results were very impressive, and I would like to pay attention to them, that this result shows that there is a significant drop in the expression of Gal-16 in breast cancer tissues. So basically, it directly indicates that Gal-16 may be considered like a tumor suppressor. And these observations actually pretty much consistent with some other reports with different type of cancers where the expression of Gal-16 expression was found to be downregulated, like a prostate cancer, colorectal carcinoma, and acute myeloid leukemia. So to look at the to confirm this bioinformatic data, we use a different type of breast cancer cells here of different molecular with different molecular subtypes, and look at the effect of the ATRA, the common inducer of cell differentiation, on the changes in expression of Gal-12. So as you see here, we have found out that the Gal-12 is significantly upregulated in response to the ATRA in MDM-B21 cells. This cell line represents triple negative B type breast cancer, and which is highly metastatic. Other cell lines what we tested in our lab, they didn't show an upregulation of Gal-12, which may depend on the ability of ATRA to induce this differentiation of these cells or their molecular subtypes. So next, the last model what we used was the model of model which is based on the differentiation of HL60 cells, which were originally obtained from the patient with acute myeloid leukemia. These cells can be relatively easily, straightforwardly induced to be differentiated into neutrophil-like cells with such substances like ATRA and MSO. Well, this process can be, this differentiation on the top, you see a different type of signs of differentiation or hallmarks of differentiation of the cells, changes in the nuclear morphology. The cells, neutrophil-like cells, they have a very specific lobular structure, shape of the nuclei. We see a drop in the nucleation, and we see increase in the expression of the markers of neutrophilic differentiation, like the NCF1 here. This is a gene which encodes the component of NADPH oxidase of neutrophils, which are responsible for generation of reactive oxygen species. So, and but when we looked at the expression of Gal-12 in these cells, we noticed this expression, but this expression was upregulated in cells which were differentiated by ATRA and downregulated in the cells which were differentiated by DMSO. And this type of finding was very interesting because it means that depending on the type of differentiation stimuli, we generate actually different phenotypes of neutrophils. And if we think about those tumor-associated neutrophils, they also have, there are different phenotypes which are called N1 and N2, which with anti-tumor pro-tumor properties. And our data actually suggest, based on the respiratory burst of this, or respiratory burst of these differentiated cells induced by PMA of FMLP, that they actually might be phenotypes which represent this N1 and N2 phenotypes of the tumor-associated neutrophils. So here we have a little bit more complex situations, because in this case it's not like we not directly Gal-12 in this case can serve as not only as a tumor suppressor, but can in case of its downregulation, it can describe the pro-tumor phenotype of tumor-associated neutrophils. So, and next, last slide, what I will to show you indicate very interesting aspect of the regulation of galectins, and they're just involvements of different type of regulation of the cells depending of where they present inside of the cells, outside of the cells. So Galactans are molecules which are secreted. Of course, they are localized in the cell organelles and cytoplasm, but they also secreted from the cells through the unconventional secretion pathway because they do not have ER localizing segment sequence. So, and we showed that for many galactans, they are secreted from the cells in so-called O-glucnag dependent manner. So basically, there is a need to decrease the amount of O-glucnag-related proteins, and this way they become secreted. For example, you see here the case for GAL3 and GAL9 very clearly. Once the HL6 is differentiated, they secrete a lot of these galactans from the cells, and same things happening if you just decrease the amount of glucose-related proteins in these cells. But this is not the case completely for the GAL12. Once the cells get differentiated, secretion of galactan 12 is blocked completely. And this blockage happening in a completely different way, O-glucnag independent way, due to the accumulation of the lipid droplets in cells. And remember, I mentioned before that this is like a very important part of the which regulates potentially the availability of galactan 12, depending on the amount of lipid droplets in cells and depending on the lipogenesis which may happen in normal or cancer cells. And this is like I'm completely here. You see my conclusions. Basically, altogether, what I would like to mention that I would like that our studies, what we are doing right now with this highly tissue-specific galactans can take more attention and can be developed in the future and will help to elaborate a new strategy for not only for like a new strategy to use them as a prognostic biomarkers and to define the function of some galactans as a tumor suppressor. So thank you for the attention. Here are the names of my students who did hard work in my lab and my colleagues for their efficient collaboration. Thank you.
Thank you so much, Professor Timoshenko. Thank you for your presentation, for having been here with us, and despite also the difference in the timetable. Thank you so much, and I hope to see you very soon. And after Professor Timoshenko, we have finished the panel list of our speakers. We are very grateful to all of you for having provided something new, for having described in more detail your research activity. And we really hope that all these topics can be important and stimulating for each of us, also to establish new collaborations or to broaden our research network. So thank you so much for your kind participation.
Thank you very much, all speakers and all participants. And we invite you for the next webinar, and we will establish a new appointment next year, I think.
Yes.
Okay.
Good.
And we are grateful for your presence at this webinar. Thank you.
And last, I will also to thank you, the organizing committee and the Bentham Science Publishers, yes, for having believed in this opportunity, in this floor, to connect people and to connect the researchers worldwide. Yes, it's true that we are two editors-in-chief of two journals, but as we are authors and co-authors of 5 papers, reviews, and so on. So we know how much it's difficult is to publish, and also to find the right journal for our research activities. So we hope that we can suggest also other opportunities. And please do not forget to register your CV, not only as a reviewer, but also as an editorial board member, for example. In addition, in each journal web page, there are specific links also to become an editor-in-chief, because Bentham is actively searching for new scientists worldwide to lead specific topics or specific journals. So thank you so much. Surely next year we will organize a second webinar, inviting other people and trying to enlarge as much as possible our network. And we wish you a very pleasant evening, and thank you again for having been with us today. Okay, thank you.
Thank you very much.
Bye-bye.
Bye.